Hello and good afternoon, everyone. Welcome to the HNRCA Monday Seminar Series at Tufts University. Today's format will be a speaker presentation followed by a moderated question and answer. Please feel free to submit questions in the Q&A box at any time during the talk. Our invited speaker today is Dr. Shingo Kajimura from Beth Israel Deaconess Medical Center and also professor at Harvard Medical. Dr. Kajimura's lab aims to understand how our body maintains energy metabolism in response to factors such as nutrition and exercise, with a focus in adipose tissue. Dr. Kajimura's research is key to understanding multiple aspects of health, including obesity, diabetes, cancer, and aging. Dr. Kajimura, your research is important to our center here as we are particularly interested in understanding links between lifestyle factors and metabolic health. And generally, your investigation of the molecular biology is key to guiding other clinical research and ultimately developing new interventions to improve human health. So thank you for being with us today. And I'm now pleased to welcome our speaker, Dr. Shingo Kajimura.
All right. Well, first of all, thank you so much for the opportunity, and also thanks for your time to listen to my talk. We moved about 2 years ago, so if I told them that we're still, I personally think that this winter is cold enough for us, but other than this, we're doing great. So what I'm going to talk about today is about brown fat, and then maybe a little more molecular, but what I'm going to talk about is why brown fat is good for you. The concept, I assume many of you, probably most of you, know about brown fat, and then for the last 40 years or so, the research essentially said that brown fat is a thermogenic organ, and brown fat is best known for thermogenesis. And then recent studies in rodents and also humans says that brown fat is protective against metabolic disease, particularly improvement in insulin sensitivity. But the question is why? Why brown fat is good for you? And then because of the best well-known function of brown fat as a thermogenic function, people, including myself, presume that this is related to energy expenditure. And then active brown fat leads to energy expenditure, and this leads to body weight loss. And eventually, as a consequence, this leads to improved metabolic health. But is that really true? Is that really this linear relationship is true or not? That's just something I want to talk about today. I'm going to spend 10 minutes or so talk about basic biology of brown fat. But many of you probably don't need this, so I'm going to keep it very quick. But the brown fat is probably one of the cell types that contain mitochondria at the highest density among mammalian cells. And actually, the color of brown comes from mitochondrial ion, so that any cell type, including cardiomyocytes or brown fat, contain a lot of mitochondria, are brown. But also, they are uniquely have a unique function. For example, besides high fuel oxidation, they have very unique machinery to make heat. It's called thermogenesis, and these features are really unique, different from any other cell types, including white adipocytes that have low mitochondria, low fuel oxidation, and function as energy storage. For the last 10 years or so, the field appreciated this another sort of the type of cells, recruitable form of brown adipocytes we call beige, which is actually there's a certain environmental cues, a subset of white adipocytes adopt a thermogenic phenotype. It's been known as a browning of a white fat, but this basic concept is that this is a recruitable form of brown adipocytes that exist in adult humans as well. So that the field of brown fat for probably last 10 years, about 12 years or so, this field is completely transformed. And one of the major reasons is the appreciation of brown adipose tissue in adult humans, and also appreciation of this significance in metabolic health. For example, I mean, earlier work around 2009 for independent groups used this FDG PET scan to look for glucose uptake and then eventually they found symmetric glucose uptake even in subjects who actually, particularly after cold exposure. And now, for example, this study used from NIH used very high sensitive FDG PET scan to detect brown adipose tissue throughout the body, particularly for supraclavicular regions, cervical, and in paraspinal, abdominal regions, et cetera, et cetera. The regional observation in 2009, which is one of those from New England Journal of Medicine, says that there's an inverse correlation between brown fat. For example, this is y-axis is a brown fat activity, and then this inversely correlate with BMI or body mass index or adiposity or maybe body weight. And then the explanation is that maybe brown fat is count for additional burning calorie up to 200 kilocalorie. Therefore, brown fat is sort of protective against obesity. That was sort of the original observation. But later, this is not as robust as people initially thought. And there's not much clear connection to body weight or adiposity per se. But what is really clear, what is really robust, is that brown fat is protective against many metabolic disease. For example, this is a study from Paul Cole and Rockefeller. They chase a large amount cohort, about 50,000 subjects. And if they compare people who have active brown fat versus people who don't have detectable amounts of brown fat, and then one of the major difference is actually let's say type 2 diabetes. If you have brown fat, those people have a lower odds ratio of type 2 diabetes by 50%. I mean, I'm sure you appreciate this. Often people talk about 5 or 10% difference in odds ratio, but now we're talking about 50% reduction odds ratio for those who have brown adipose tissue. So this number is pretty significant and very robust. And also protective against dyslipidemia, coronary artery disease, cardiovascular disease, etc. And particularly protection against type 2 diabetes is replicated in many other independent studies as well. What is very interesting to me and then to us is that this protective effects against type 2 diabetes is independent of body weight. What I mean by this is that in this study, if they stratify the subject based on body mass index by BMI over 30 or mid-range or low below 25, you still see significant reduction in prevalence of type 2 diabetes if you have brown fat. So somehow it's clear that brown fat is protective against, let's say, type 2 diabetes, but the effect is independent of body weight. Another interesting thing is that a human, as I alluded earlier, there's a sort of recruitment process. So there's an inducible form of thermogenic cells exist in adult humans. And in one of the really remarkable studies, again, from studies from NIH, show that, let's say, they actually track people who did not have appreciable amounts of brown fat after acute cold exposure. They don't have any detectable glucose uptake in the supraclavicular regions. But then they let those subjects to chronic cold exposure, chronic activation beta-adrenergic receptor agonist. And up to 3 weeks later, essentially most subjects regain new brown fat. And also this is accompanied by improved insulin sensitivity. So this is really exciting to the field that, first of all, brown fat is recruitable in adult humans, and this is accompanied by improved insulin sensitivity. So it's not, so now the question is, why brown fat is good for you? And then before that, I'm gonna let me talk just background or sort of dogma as to how cold stimulate brown fat thermogenesis. This is very oversimplified scenario, but just the background knowledge. So what is being very well appreciated is that a cold is sensed by sets of neurons in the brain, which stimulates sympathetic nerve the release of norepinephrine from the nerve terminus of the sympathetic nerves. This binds to beta 3 adrenergic receptors, which trigger lipolysis, but also this leads to activation of brown fat. And then specifically, this is a molecule called uncoupling protein 1, which exists in the inner membrane of brown fat and in beige fat. This is the UCP1. I'm sure many people heard about this. This is a uncouples proton gradient from ATP synthesis. So instead of making ATP, this UCP1 is a channel or carrier for proton, so they reduce the mitochondrial membrane potential without making ATP. So this, according to Joule's law, right? Heat production is proportional to resistance. And in this circuit, UCP1 is really the resistance circuit. Okay, so that's how people appreciate it. So for the last probably 30 years or so, people assume that this is the only thermogenic protein that mammals have, and a UCP1 is really responsible for thermogenesis. So accordingly, the dogma that I mentioned earlier is that as to how brown fat improves metabolic health is through thermogenesis by UCP1. And then this thermogenic activity through UCP1 leads to increased energy expenditure at the systemic whole body level. And eventually this leads to body weight loss. And then this eventually leads to metabolic health, insulin sensitivity. But the data that I'm going to show you today, we suggest alternative processes. And then I'm going to emphasize the role of branched chain amino acid in this context. But just for historically background, I think let me begin with this paradox that a field appreciated for the last 20 years, which is about inconsistency of metabolic phenotype between UCP1 knockout versus brown fat ablation mouse. So here, 1993, a work by Jeff Flyer, Brad Lowell, developed this mouse model where they ablate brown fat by overexpressing diphtheria toxin. So this is a diphtheria toxin-mediated ablation brown fat. Brown fat defective ablation mouse eventually become obese and in room temperature. And a couple years later, Leslie Kozak and Sven Nenabak actually generated the first UCP1 knockout mouse. And then they show different phenotype, which is the mouse, UCP1 knockout mouse are cold sensitive, but they're not obese, as long as they keep the mouse in room temperature. And eventually Barbara Cannon, Jan Nedergaard said that UCP1 knockout mouse can be obese if you keep them on 30-degree cold thermal neutrality. But the fact is that phenotype of UCP1 knockout mouse and brown fat ablation mouse are different at room temperature. So something is missing. The caveat of this experiment, particularly this study, is that they use diphtheria toxin, which trigger massive inflammation. And particularly in the context of type 2 diabetes, this is not a best model to study it. So but at least in body weight regulation, the phenotypes are different. So then let me tell you how we get into this from our perspectives. And back in old days, since I was a postdoc in Bruce Spiegelman's lab, we tried to understand brown fat development, where cells come from, what are transcriptional regulation of brown fat. And then we identify one of the sort of key transcriptional circuits called PRDM16 complex, which is a PRDM composed of PRDM16, PRDM3, and then one of the methyltransferases called EHMT1. This complex regulates the cell fate decision of the progenitor cells, either go to brown fat or in this context, skeletal muscle. So because of that, then we're able to generate cell type specific knockout. In this case, we knock out EHMT1 in a fat specific, progenitor cell specific manner, and then we're able to generate brown fat ablation mouse model without using diphtherotoxin. Okay, so this is a very this actually allow us to ask about the role of brown and beige fat in the regulation of glucose homeostasis. And in that time, when we did in this about 2013, when I started my lab in UCSF, what we learned is that at that time those mouse have defects in thermogenesis, which is consistent with our other research. But what's also we found that those mouse, if you ablate brown and beige fat, by knocking out this gene, then the mouse develop severe hepatic insulin resistance. Okay, this is the Western blot for phosphoric 18 in response to insulin. So insulin stimulates phosphoric 18 wild type, but this brown fat ablation mouse have reduced insulin sensitivity, insulin signaling, and eventually suggest that idea that maybe those brown fat ablation mouse have insulin resistance in the liver. Later, we also show that this fat adipose tissue have fibrosis, inflammation, and these are somewhat related to the beige fat. And also these phenotypes are consistent with fat-specific knockout for PRDM16, which published by Bruce Spiegelman's lab, Patrick Seale's lab, etc. So, but then this argument that I told you came out. This doesn't make any sense when you think about UCPM, because UCPM knockout mouse are not diabetic. They're not insulin resistant at room temperature. They don't develop any fibrosis and inflammation in adipose tissue. So clearly that the brown fat is doing something different from UCP1. Then how we test that sort of UCPM independent action of brown beige fat. And then around the time, we were able to make some really unique mouse model, which is that we generate mouse that express PRDM16 to genetically make more brown, in this case, beige fat in their subacute adipose tissue, and cross this with UCPM knockout mouse. So this mouse model have really sort of strange setting, I mean, very artificial in the sense that they lack UCP1, but they have a lot of beige fat in their subacute fat. And then this actually allow us to test how the metabolic phenotype of beige is dependent upon UCP1 or not. And then this is a pretty shocking data to us. Essentially, here's a GTT, glucose tolerance test of mouse. These mice have a lot of beige fat but lack UCP1, or legitimate control mouse, which is UCP1 knockout mouse. And then the data sets that essentially, if you have beige fat, then the mice are more tolerant, gain high tolerance to glucose, regardless of UCP1. And then same thing, it's insulin tolerance test, so that of these mice with more beige fat are insulin sensitive, even though they lack UCP1. So the reason why we're really shocking is, again, is that because of the fact that we presume that the brown and the beige fat is good for you because of thermogenesis by UCP1. But the data that we found suggests that UCP1 is not required. UCP1 is not required for the anti-diabetic action of beige fat, particularly for improved metabolic health. So this actually was, and with that data, I decided, we really have to figure this out, why that is, and then why brown and beige fat it doesn't really need UCP1 to release the insulin resistance, improve the insulin sensitivity. What are the mediators are? So that's the sort of the background, historical background as to why we start working on UCP independent pathways that regulates metabolic health. Okay, so the concept, I put that concept in about 2015, way before actual data comes along. But we essentially, for the last 7 years or so, we really tried to understand the role of brown fat beyond thermogenesis. Just make sure that, don't get me wrong, thermogenesis is I'm not saying thermogenesis is not important, okay? Brown fat thermogenesis is important. UCP1 is important. And then particularly now UCP independent thermogenic pathway exists. So thermogenic aspect is clearly important for heat, for regulation, energy homeostasis, energy expenditure, or regulation of cold tolerance and temperature regulation. But what I mean by this is that this thermogenic function may not necessarily is directly links to metabolic benefit, particularly insulin resistance, insulin sensitivity. And then, so there are a lot of the sort of non-thermogenic function. It's not a clear cut. Some of them are related to thermogenesis, so I shouldn't say the clear cut, but there are multiple functions that a field, including ourselves, actually start to appreciate. For example, brown beige fat secrete unique cytokines called cytokines. Particularly beige fat, browning of white fat is accompanied by dynamic tissue remodeling in adipose tissue. Enhanced angiogenesis, inhibition of fibrosis, inflammation, some immune cell recruitment is involved. What I'm going to talk rest of my time is about metabolic sink for branched chain amino acid, glucose-free fatty acids, et cetera, et cetera. So this is the area that I'm going to talk for the rest of my time. So how we got into this, why we got interested in branched chain amino acid, is from completely unexpected. So here's a transcriptomic analysis of mice that I told you, which is that the mice that have a lot of beige fat by overexpressing PRDM16 but lack UCP1. Again, I did this UCP1 knockout mouse as a control, and even with this condition, even in absence of UCP1, if mice have more beige fat, they are protected against glucose intolerance and also insulin resistance. So why? And then we did transcriptomic analysis, and number one hit, metabolic pathway that upregulated in this beige fat, regardless of UCP1, is active catabolic pathway of branched chain amino acid. I'm sure many people know a branched chain amino acid. So, for example, BCAT2, which is a mitochondrial localized deamination enzyme, BCKDH, and then that downstream pathways. And this was somewhat interesting, but it wasn't really the top priority at the time. But regardless, I thought that this is somewhat interesting, so we decided to follow up by using this tracer, flucoclovine, which is a radioisotope labeled leucine analog, where we can inject this into to tail vein and through circulation and see which tissue actively uptake BCAA. It turns out the brown fat right here, which particularly after cold exposure, brown adipose tissue actively uptake BCAA. And this is a quantification where in the brown adipose tissue from thermoneutral acclimated mice versus cold exposed, and clearly brown fat actively uptake BCAA on a leucine analog into the brown fat. Same thing for inguinal-adipose tissue, suggesting that both brown and beige actively uptake BCAA. And also, I should mention that there are several other labs actually made a very similar conclusion. For example, Zolta Rani's lab and Chris Newgard, Sam's labs, and etc. And then not only in mice, but also in humans, this similar observation were made. For example, here we collaborated with scientists in Japan. They have a cohort of composed of people who have active brown adipose tissue based on FDG PET scan, or people who did not have active brown fat. So the brown fat high versus low. We ran metabolomics of the circulation before and after cold exposure. And one really interesting observation is that valine leucine levels are reduced in the circulation after cold exposure. But this is selective to people who have high active brown fat, but this is not seeing people who have no brown fat. So this suggests to us that brown fat promotes BCAA clearance in humans. And then again, this is very similar observations are made by other scientists, for example, Aaron Cypress's lab and Saito's lab, and et cetera, et cetera. So it's something about BCAA and brown fat. This caught our attention for several reasons, but number one is that it's been known for 20 years or so, or even more, that there's a clear, strong, tight correlation between elevated circulating BCAA levels and insulin resistance in human cohorts. For example, a study from Birmingham cohorts, and then more recently, though, Chris Newgard's lab and others show that this is the data from Chris Newgard's paper in 2009, showing that if you plot this HOMA value against BCAA-related metabolism, there's a clear correlation there. So that somehow BCAA is considered to be a biomarker that is start to elevate in type 2 diabetes or insulin resistance. And then the fact that the brown fat promote clearance, so the brown fat is activated, then probably uptake BCAA to clear BCAAs in a circulation, which is sort of consistent with that idea. So I thought this is potentially interesting. But here's the problem. Here's really the problem. And then we puzzle for years, and even nowadays, is that the BCAA can be good or bad, depends on which tribes you're in or which field you are in. For example, in the diabetes field, BCAAs, branched amino acids, are considered to be bad guys because of the epidemiological studies that, again, I mentioned earlier. Circulating BCAA levels are seen in obesity or type 2 diabetes. And there are some studies in animals that BCAA supplementation together with high sucrose or high fat diet, it actually causes insulin resistance. And therefore, BCAA is considered to be a bad guy in terms of the glucose homeostasis. On the other hand, in nutrition field, I'm sure many of you in the audience aware that BCAA supplementation is known to enhance energy metabolism and also improve muscle performance, particularly after intense exercise, and good for muscle recovery. So the BCAA, a lot of athletes and then bodybuilders actually take BCAA as a supplementation. So the question is, again, why BCAA can be good or bad? right? This is really not clear to me. And I'm going to get into that topic later. The second paradox that particularly nowadays, we more recently think about this is that there's BCAA is really how cells actually use BCAA, right? BCAA is obviously the essential amino acid, and BCAA is presumably oxidized in the mitochondria, and the carbon getting to the T-state cycle as a carbon fuel. But the reality is that, for example, this really elegant study by groups in Princeton, for example, map this quantitative flux of circulating metabolites in different tissues and asking what are the major carbon fuels in each tissue. For example, brain, clearly the glucose is the major fuel. Glucose accounts for about 70% of carbon into the T-state cycle, and in other tissues, and then, for example, lactate, glutamine, etc., etc. But BCAAs, as shown in green, never be the major carbon fuel in for example, brown fat or other tissues. So clearly, BCAA is not it's not just a fuel that burn in a T-state cycle. So these are the sort of paradox that we had for over years, and then I hope our data will tell you or explain some of them. Not all of them, so at least some explain some of those paradoxes. Okay. Now, so BCAA again back to this, is an essential amino acid. And essentially means that it has to come from food. And BCAA can be catabolic or anabolic, depends on how they use it. And then again, BCAA is essential amino acid, so that every single cell have to use BCAA for protein synthesis, right? Particularly pancreas, they have to make a lot of insulins and etc. So they take BCAA a lot. And also BCAA is important activator of mTOR pathway, so that this is occur in the cytosolic fractions, and it's in the anabolic action. But in many metabolically active organs, including brown fat, BCAA is actively uptaken in the mitochondria and getting to the T-state cycle or used for something else. Because the first step of deamination, called BCAT2, localized in the mitochondria. So the question is how cells decide to use for anabolic reaction or catabolic reaction. More specifically, how they bring BCAA into the mitochondria. But this is never be clear, because of the mitochondrial transporter for BCAA is not identified. Just a reminder, mitochondrial inner membrane is very tight, impermeable. It doesn't even penetrate proton. That's why they can make proton gradient across the membrane. This means that every single metabolites that get into the mitochondria need designated transporters or carrier proteins. And in the fact that we found the brown fat actively oxidize BCAA in the mitochondria. We search for this mitochondrial inner membrane carrier for branching amino acid. And then a couple of years ago, we found a candidate. It's called SLC25A44. This is an orphan carrier in the mitochondrial inner membrane. If you delete this protein in the mitochondria, then mitochondria fail to uptake BCAA, valine and leucine. So, suggesting that this SLC2044 is responsible for, at least require for, a BCAA uptake into the mitochondria. So this allows us to ask how, to allow us to manipulate the BCAA utilization within a given cell type. Later, we actually did a little bit more biochemical studies. For example, here we reconstituted the BCAA transport in a cell-free system. It's called liposome. We made protein, and then put into the liposome, and then in fact that this protein is mediates leucine uptake into the liposome, which is good. And if you block this MBC, we essentially, SLC 2544 is a terrible name, so we call it, for easiest, we call mitochondrial BCA carrier, or MBC. And if you knock out MBC, then the BCA oxidation in the cell is reduced, which makes sense. But also, if you block this, then since this blocks or impair BCA flux in the circulation, so the knockout mouse have elevated BCAA level in the circulation. Here's the fasting BCA levels of control mice versus whole body MBC knockout mouse. So that plasma BCA levels in vivo is higher, which is consistent with the notion. How this protein works, just for brief summary, what we learned so far is that this MBC from a former cluster with, let's say, thymine carrier or a dicarboxyl carrier, a phosphate carrier in the mitochondrial inner membrane. So this inner membrane, and then this explains how this carrier protein is coupled with BCA oxidation. For example, thymine carrier is needed for activation of BCKDH. Dicarboxyl carrier is important for increasing TCA flux here. But also, this tells us that MBC is coupled with glutamate carrier, SLC2522, and it's required for deamination step of BCA, because again, BCAT2 deaminate nitrogen, right, from the BCAA, transfer to an alpha ketoglutarate to create synthesis, to initiate the synthesis of glutamate. So this explains beautifully that two things: MBC is somehow coupled with BCA oxidation, but also required for nitrogen donors. And I'm going to get to this point later. Now, what is the biological role or significance of this transporter MBC? So Takeshi Unishiro actually generated a whole body CRISPR knockdown mouse called CRISPR-I MBC mouse, and it was gratifying for us to see that this mouse whole body knockdown on MBC leads to glucose intolerance based on GTT here, and also insulin resistance. ITT showed that the knockout mouse have reduced insulin sensitivity compared to control mouse. This is a whole high-fat diet, room temperature. So meaning that this knockout mouse defect in BCA flux in the mitochondria is sufficient enough to cause glucose tolerance and insulin resistance, which is good. But the caveat is that this transporter is expressed in many, many other cell types, including muscle and in other tissues, so that we decided to knock out this in one by one, but tissue-specific manner, and identify the responsible tissues. So Tony Verkerke, he's a postdoc. He took over this project. He generated brown fat-specific knockout mouse, MBC knockout mouse, and then challenged them to cold. I mean, idea is that BCAA oxidation is required for brown fat thermogenesis, which is true in embryonic stage or prenatal, postnatal rarity pups. They're clearly important, but also in mice and adult mice. And then first experiment he did was to cold challenge of those mice, and it was very disappointing to us that there's no phenotype. Brown fat specific loss of mitochondrial BCA carrier did not affect adaptive thermogenesis. Tony did a careful examination of whole body energy expenditure using metabolic cages, different temperature. Essentially, there's no significant difference. There's really subtle difference in cold, but essentially pretty minor. So, and this is also true for a fat-specific and adiponectin-free knockout MBC. So it was disappointing, but would make a lot of sense now, because the MBC is important for I mean, it's true that brown fat uptake BCAA, but it's not a major carbon fuels, such that if lack of BCAA oxidation can be compensated by active uptake of glucose or fatty acid. So lack of BCAA is not necessary or not sufficient enough to cause energetic defects in brown fat in vivo, particularly in adults. Again, in mice in the early phase, like newborn pups, BCAA is important, but in adults, MBC is not required for adaptive thermogenesis. But it was also, on the other hand, it's fascinating that they are sufficient enough to cause insulin resistance. Here, NACA mouse have insulin resistance, which is really robust and also consistent with adiponectin NACA mouse. So the question is, which tissue is responsible for that? It turns out actually the liver. The liver, if you blot phosphorylated AKT in response to insulin of control versus brown fat, specifically MBC NACA mouse have reduced insulin sensitivity or signaling. And then we also did phosphoproteomics and it confirmed that some of the protein insulin response signaling phosphorylation is impaired in NACA mouse. Then question comes to this. How does impaired BCAA flux in brown fat leads to hepatic insulin resistance without affecting energy expenditure and thermogenesis in brown fat, right? Remember that I told you that brown fat is known for thermogenesis, but here is a very, very unique examples where brown fat BCAA fluxes down, but thermogenic capacity itself is not really changed, but the mouse developed insulin resistance. So how can we explain that? It took us a long time, but first observation, initial hypothesis was that, okay, BCAA is also known for lipogenesis, so maybe this leads to hepatic steatosis in the liver. So 20 major triglyceride contents in the liver of control versus brown fat specific MBC NACA mouse, no different. Disappointing. But he also made a very interesting observation that protein carbonylation is higher in the liver of NACA mouse, suggesting that some oxidative stress is higher in the liver. Interesting, but we had no idea what it means. Then we did this very, eventually this actually told us a lot, which is about nitrogen tracing. Again, I told you this BCAA is not a major carbon fuel. So we ask how cells use nitrogen from BCAA. So we use nitrogen tracer veiling of knockout versus controls, and then makes sense, veiling is down. But also we found that the cells have defects in or lower level of labeled aspartate, glutamate, and alanine glycine. And in one point, we talked to collaborator Duke, Phil and Rob, and about this data, and then they say it must be glutathione, because glutathione is actually made from glutamate, glycine, cysteine, and both of them are reduced in the cells. In fact, then brown fat cells actually have reduced glutathione, labeled glutathione from BCAA. It turns out that BCAA is a key nitrogen source for the synthesis of glutamate, but also as a side product, eventually glutathione. With that data, now we look back all the mouse samples that we have. For example, we measure serum level of glutathione or GSSG in control versus brown fat specific MBC. Serum level glutathiones are down. Brown fat glutathione levels are down. And also liver glutathione levels are down in the NACA mouse. This explains why liver has higher oxidative stress. It's probably glutathione. If that's the case, then can we test that idea by supplementing glutathione into knockout mouse. So here, Tony did this experiment. This is another cohort of control versus brown fat specific MBC knockout mouse. Then again, they're consistent with our observation. They're insulin resistant based on insulin tolerance. We supplement these mice with glutathione for up to 10 days. And then after 10 days of glutathione, restore insulin sensitivity of the knockout mouse. Conversely, this is actually an experiment where we depleted glutathione in mice by injecting this inhibitor for the glutathione, the synthesis enzyme, BSO. And then here, the serum level GSH is down as expected, which is being replicated in a number of studies. BSO treatment, reduced serum BCAA, but this leads to elevated BCAA levels in the circulation, which is interesting. But this is sufficient enough to cause insulin resistance based on insulin tolerance test. Insulin tolerance tests of BSO-treated mouse have impaired insulin sensitivity, so that it suggests that depletion of GSH elevated BCAA levels, but also that is sufficient enough to cause insulin resistance. So this data tell us that maybe BCAA catabolism is somehow coupled with GSH synthesis in brown fat. So one experiment is how it's regulated. So here's one experiment we did. Tony fed mice on regular chow versus high-fat diet for 2 different stages, 4 weeks or 12 weeks. We harvested tissues, brown fat, inguinal fat, liver, different tissues. We measure oxidation, leucine oxidation, essentially BCKDH activity, and it shows that brown fat BCAA activity is dramatically down after short-term high-fat diet. Somewhat similar, this is true in inguinal fat, but no difference in other tissues. So the BCAA catabolism in brown fat declines in obesity. When we purified mitochondria from these samples, we ran quantitative metabolomics, proteomics, we call mitochondrial proteomics. Many of the enzymes are downregulated in mitochondria from brown fat, for example, BCAT2 and many others. When we look those then, pathway analysis shows it's very dramatic. Many of the pathways are dominated by BCAA catabolic processes. But this is not just a general decline of mitochondria, because there's no difference in, let's say, TCA cycloenzymes or many others. Mostly the downstream catabolic pathway of BCAA is down. In this context, we measure glutathione levels, and in fact, that brown fat from high-fat diet mouse have reduced GSH levels, so that declined BCAA catabolism is coupled with reduced GSH levels in the brown. Lastly, we collaborate with the Takeshi Unishiro and Saito's lab in Japan, where they also measure BCAA levels in circulation humans before and after cold exposure. And then what they found was that the GSH levels are somewhat up. It's not dramatic, clear up after cold exposure, but what is clear to us is that after cold exposure, GS level in the circulation is higher in people who have active brown adipose tissue as compared to those who don't have active brown fat. So somehow active brown fat is coupled with elevated GSH in humans. Together, here's a conclusion that from what we learned so far. I told you about the paradox, why BCAA can be good or bad, depends on the field. What we learned, our interpretation of this paradox is that maybe elevated circulating BCAA levels are likely a diffraction of reduced BCAA flux and or mitochondrial BCAA catabolism, for example, in brown fat activity. So if you have, it's not about high BCAA is a causal aspect. Rather, we consider that this is the elevated BCAA levels in circulation is a consequence of reduced BCAA flux in peripheral tissues. And for example, if you have reduced BCAA flux in brown fat, this leads to elevated BCAA levels in the circulation. Second is, our BCAA just carbon fuel? It's clearly not. For example, in brown fat, BCAAs are major nitrogen source for the synthesis of glutamate and glutathiones. So that in fact that brown fat actively uptake BCAA, not for a fuel for carbon, but probably used for the synthesis of amino acid like glutamate and glutathione. That's what we think is. So big picture, the first question of why, I told you this, why brown fat is good for you. Our data suggests the following. For example, we at least thought the UCP1 is responsible, but that's not the case. UCP1 is dispensable for metabolic benefit of enhanced brown beige fat. Clearly, this is about UCP independent mechanism. I did not talk about thermogenic pathway. For example, creatine cycling or calcium fetal cycle. It regulates energy expenditure, and this is also true. But what is really important for the insulin sensitivity particularly is about the clearance of metabolites and maybe brown fat derived factors, at least in the context of BCAA catabolism. BCAA derived metabolites, for example, glutamate or GSH, could be important. And then clear the sort of the flux or BCAA flux is actually linked to insulin sensitivity rather than enhanced thermogenesis per se. All right, so I'm going to close my presentation by thanking our lab members, particularly Tony Verkerk. He actually led the work on, he's right here, he led the work on BCAA catabolism in the context of insulin resistance. And then Malia is a graduate student who helped Tony. Dan One is a staff scientist. We have now mass spec running metabolomics, and then and also our former trainees, Takeshi, Zach, and collaborators, and of course funding sources. So with that, thank you very much for your attention, and thanks for the opportunity. And I'm happy to answer any question you may have.
Thank you, Dr. Kajimura, for a very good presentation. Let's move to the Q&A. A reminder to attendees, you can type questions in the Q&A box or use the raise hand feature in Zoom to ask any question. Okay, so our first question is from Larissa Constantino. Could BCAA supplementation increase the risk of insulin resistance? May the increased BCAA level in type 2 diabetes be just a consequence of metabolic impairment?
Yes, that's what I said. So the BCAA is elevated BCAA in the circulation deflects sort of impaired BCAA flux as a body, and also the impaired catabolic pathway, including brown fat. So that being said, the study, some of the nutrients BCAA supplementation studies suggest that if you combine that BCAA supplementation together with high sucrose diet or high fat diet, that actually enhance insulin, the risk of insulin resistance. So that probably explained that maybe it's a combination to stress them the mitochondria or a catabolic pathway, then that may be increased. So the short answer to your question is that maybe BCAA supplementation is not a good idea for those who have high insulin resistance or type 2 diabetes or reduced BCAA flux. But as we all know that a BCAA supplementation can be beneficial as long as you're healthy, and if you have active brown fat and also can catabolize BCAA and in a sense the skeletal muscle brown fat. So it can be good or bad, depends on the more metabolic status.
Thank you. Next question is from Shangdong Wang. Great work and presentation, thank you. I was wondering if beneficial or detrimental effects of BCAA are related to gut microbiome, which may affect BCAA catabolism and their metabolites, BACC flux, or BACC catabolism in brown fat?
Yes, there are studies actually done by groups, including Chris Newgar said that microbiome, right, is actually metabolites BCAAs in different ways, and that also implicates that. So clearly, we don't really study the involvement of microbiome in this context, but I assume that's one of the important regulators.
All right, thank you. Andrew Greenberg asks, is diet the only source of BCAA?
Great, Andy, which is actually, it's a great question. It's not. I mean, BCAA is considered essential amino acid, but clearly certain cell types, for example, cardiomyocyte or other cancer nowadays, there's BCAT is a reversible process that certain cell type can make aminate, put a nitrogen on to keto acid to make BCAA. So it's being sort of implicated in heart failure and other contexts. Now, how important that is in physiology is not clear. But certain pathology, certain cell type can generate BCAA from keto acid. Of course, the other one is the muscle degradation, right? The muscle, if you have muscle extreme fasting or muscle degradation leads to protein synthesis, including BCAA. So I'm not saying this is clearly dietary is major one in physiology, but other context, other way of making BCAAs.
Okay, Sheldon Rowan asks, circulating glutathione drops significantly with aging. Have you explored the impact of aging on oxidative stress in brown beige adipose tissue? Yeah,
this is an amazing question as well. We as soon as we saw that, the one idea is related to aging. For example, it's been known that brown fat prevalence dramatically decline with age. And what is the causal relationship between the two? That being said, glutathione, as you know, is generated in many other tissues, and brown fat can do, but how much of that is a major contributor is not clear. So that's exactly what we're doing. What is the contribution glutathione synthesis in the brown fat as opposed to other tissues? But a biological context in this area is probably aging, and I love to have inputs and suggestion from you guys.
Great. So that was our last question. I think I'm just checking, or if there's any comments after your answer to the last question. Just want to check that we're all set. Okay, that was our last question. So that concludes the Q&A, and that concludes the seminar. Dr. Kajimura, thank you for your time today. It was great to see everyone in the audience. Thank you for attending, and have a great day.
Thank you very much. Great. Thanks. Bye.